I frequently attend this meeting. It's a very small setting. It gives good chance for discussions. So before I get into the crux of the topic, PBPK, how many of you are familiar with it? Okay, so it's half and half, I guess. PBPK, physiologically based pharmacokinetic modeling, the modeling that you have seen before by the previous speaker here, that is where you describe the data, you collect the data, then you try to understand the time course of the concentrations and sources of variability and so on based on the data. But physiologically based PK modeling is about building the model without the data. I mean, there's data, but without the data, based on physiologic principles, the blood flow, the size of the organ, the partition coefficients based on in vitro data. This is not really using, per se, observed data from clinical trials, but you would like to use the model to predict the clinical trials, let's say, pharmacokinetics in patients or subjects without having to do the clinical trials. That's the simple definition of PBPK. Originally, when Shirley asked me here to speak, I gave this title, but then I realized I should have really made it rational application. So I will paint a 30,000-foot view for the benefit of the half of the audience who are new to this approach, and the speakers who are after me are a lot more experienced in terms of applying PBPK hands-on experience. So I have that advantage being going first. I can be vague. There are, in my opinion, 5 areas of application of physiologically based pharmacokinetics. The first one is drug-drug interactions, and I think that is what you will hear from the next speakers. I would like to have something clarified upfront. This is application to drug development, meaning your approach, PBPK, for example, or any other, is a tool. You're not going to get a decision out of that tool. You're going to get some guidance, some results, inferences that you're going to use with pragmatism, the domain knowledge, the clinical perspective, and a bunch of other things to finally make the decision. So this is giving you a piece of a bigger puzzle, which is quantitative, which is objective. But there are a lot of subjective elements that also play a role before you make your final decision. So drug-drug interactions, why do we need that? These are surrogates for potential safety concerns that may occur in the clinic, but you will not be able to do efficacy safety trials. We have agreed as a community to look into pharmacokinetics in a very well-controlled manner and then see if there is a need for dose adjustments or some kind of labeling instructions what to do when you have the potential co-administration of other drugs. Especially in the area of antivirals, you have plethora of potential drug-drug interactions. In fact, that is I think 40% of your drug label, which most people don't read. And the drug development focus or the question for drug-drug interactions is the following. We are not interested in detecting teeny-weeny effects. There is no interest in measuring whether a drug co-administered is going to change it by 10%, 20%, even 50%. We're talking about doubling, tripling. That is when it gets our attention, meaning you don't have to be extremely precise or accurate. That I really want to exclude, not include, exclude a big effect. that I should be worried about, and maybe I have to do something about it in terms of labeling. That's the drug interactions piece. Biopharmaceutics, when we'll go into that the specifics. But biopharmaceutics is more about changing formulations. What do I do if I have to change the manufacturing site? Is there a role for physiologically based PK modeling to help the sponsors? Because that's a very big ticket item, in fact. And there I am concerned about small effects. So that is where I need a model, if I have one, to accurately predict for me the influence of changing of different excipients, the influence of changing different manufacturing processes. So that has to be accurate, precise. Bioequivalence, same thing. If I have to substitute an in vivo bioequivalence study with a PBPK model, I want the model to be precise, because I am interested in figuring out not more than 5 to 7% difference on an average. Otherwise, you won't meet the bioequivalence boundaries. Special populations where I cannot access pharmacokinetics, pregnancy, fetal exposure, exposure through breast milk. Those are studies that I may or may not be able to do. They are very challenging. So in those cases, PBPK can be useful. Medical countermeasures. These are for medical countermeasures are when there is a untoward event, radiation exposure, or chemical agents, the general public are exposed to it. How do you determine exposures and efficacy and safety? You will not be able to do clinical studies. So you base what's called as the animal rule. That's the basis. But then PBPK can have a role in understanding whether the drug reaches a particular site or not, where the action is intended to be. Now let's look at drug-drug interactions. There, in my opinion, are a couple of places where drug-drug interactions are useful. One is planning. I really think this is the single most important use of PBPK in drug-drug interactions. Sponsors will need to plan the potential drug-drug interaction studies they ought to conduct upfront in their drug development program. So there are a couple of things. One, anticipated effect. You don't have to be extremely precise, as I said before, but you need to get a feel for what you're expecting if you do an in vivo study. The second is when this is important to be done for planning, because you have to write your protocols for your patient studies, and that protocol will have inclusion-exclusion criteria, and some of them you have to decide whether to include patients on certain drugs in these studies. How do I plan that? I would plan that using these types of predictions, because you don't have to do all the in vivo studies upfront. So that's the planning part, which I think is the single most valuable application of PBPK. The second is when you submit the new drug application, where you would want to ask for waivers based on the projections, saying that yes, this is a CYP3A4 substrate, but it's not the prediction. It doesn't seem to be clinically meaningful, so I ask for a waiver, and then you can negotiate with the FDA whether you want to put that in the label or not. And I think that's still evolving. If you can put dosing recommendations based on PBPK, perhaps the next speakers will speak to it. Then let me show you an example. This is a work that we have done. Buprenorphine. How many of you are familiar with that? Yeah? Good. So buprenorphine is approved for predominantly to deal with opioid dependence. And there is there are sublingual, IV, and oral formulations available. Now, there are subcutaneous formulations that are coming up, which are long-acting. So you don't have to dose every day, because in these patients, dosing adherence to buprenorphine is a challenge. So by having monthly injections or so on could improve the clinical outcomes through better adherence. Now, buprenorphine's PK is beaten to death. It's a CYP3A4 substrate. It's also metabolized by UGT1A1. But CYP3A4, let's talk about that. And so then what do we do for subcutaneous? Do you want to do another drug-drug interaction study? That sounds silly. And it cannot be as worse as sublingual. It has to be a notch or 10 notches below sublingual drug-drug interaction potential. So what was done here is we have combined some biopharmaceutics principles and deconvolution to understand the absorption profile after subcutaneous, and then applied PBPK to project what happens in the presence of inducers, rifampin, as well as ketoconazole inhibitor. And then you see that if you cannot see big difference in the two curves, the dotted line and the solid line, there is nothing for us to talk about. The label, I can write a label saying there is no interaction that needs dose adjustment. So there are two sides of this coin. On one side, scientific, the other side, business. Scientific. I know that there are two predominant pathways for drug interactions. One is through systemic clearance, the other one is through bioavailability. I can confidently say that the most of the drug interactions that get patients into trouble are not because of systemic clearance, it's because of bioavailability. That is a huge effect, and it's very easy to detect that. For buprenorphine, predominantly, the effect is due to bioavailability, boosting of bioavailability. You know that concept very well in this crowd. So when you shift to subcutaneous, that is off. I don't have to worry about effect on bioavailability. So really, is there a question? I think we don't need to do anything. I don't even think we need to do this analysis. That's the scientific part. But then the business part. But still, the sponsor, for example, if there was a sponsor who is doing this, they still have to submit something to the FDA to substantiate labeling. If this is what is going to get them off the hook of an in vivo study, why not? I don't know. I am split between these two, but I hope my point is clear. So if I can get away without the in vivo study, I will do this kind of stuff. If I have to do it 10 times, I will do it to avoid an in vivo study. But I really think we don't need it, in my opinion. So then the point I'm trying to make is we have to be prudent in where we apply PBPK. It's not something we should be mixing in water and ingesting on a daily basis in general public. There has to be some thought. There has to be some responsibility in deciding which are the cases which really benefit from PBPK, because it is not a solution for every problem. Because some of the problems we have pharmacokinetics, rich knowledge that we have accrued over decades. We cannot ignore all that and then be excited about just a new tool because it's new and sexy, but ignore the abundant clinical pharmacology, pharmacokinetic principles that we have accrued over the years. All right. The other, which currently we don't think about and which is so relevant for antivirals, is bedside. Although during drug development you would have worst case scenarios, you may have done studies with ketoconazole. What about itraconazole? What about some other moderate, mild inhibitors? What do I expect? And this is where I think, again, there is a choice. One is vacuum right now. So I guess depending upon the day, or we can have a little bit more scientific approach of using this PBPK approach to guide the pharmacists and the healthcare providers in giving them a hint. Is it a big effect that they should be worried, or it's okay, or monitor some kind of sign? That's where I think there is going to be, there is a big need for that. But that brings us to a problem. The market size for PBPK is not big. So you have one or two software packages, but they are commercial packages. As long as we have only commercial packages that a common pharmacist cannot access, you will never be able to have this dream. Impossible. So we have to have an open source software that can be accessible to these hospitals and hospital pharmacists. That is something we'll have to invest in. Biopharmaceutics. What do I mean by biopharmaceutics? When you have a formulation change, if it's so there are 3 types of changes. If you're not familiar with it, the only thing that we would care about is major changes, like manufacturing changes or a big time excipient change, it's more than 5% and so on. Then you either have to establish an IVAVC, in vitro in vivo correlation, like the same thing you do with KI versus in vivo intrinsic clearance, or you do an in vivo bioequivalence study. And you don't want to do that, or people don't want to do in vivo studies as much as possible. So you could use PBPK to understand or correlate what happens with the different excipients in terms of their effect on absorption and bioavailability in vivo. If we can build that, then you can make a case for substituting or waiving an in vivo study. Then there is also this notion, and I have seen, I used to be at the agency once upon a time, but now I see it from distance through binoculars. But this is not about FDA. I've seen other agencies do this. They're asking companies to use PBPK approach to project food effect and alcohol effect. This is 40% alcohol. There's nobody I know who drinks that, and I deal with students on a daily basis. So we have to be realistic in expecting what can be done. Just because it is PBPK, it's a machine, it doesn't talk back, you can do whatever you want with it. So I caution that we have to be prudent. And in fact, in my opinion, we do not know the methodology and we have not built a consensus on how to use PBPK in terms of biopharmaceutics. Because remember, as I said, we are interested in small effects. Now, an example I'll tell you in terms of bioequivalence. There are several bioequivalence challenges, especially for products which are not systemic. They are given locally, but they could have a systemic effect. And establishing bioequivalence for those products is challenging because you will be required, I would say, it's guidance, so to do a clinical study. And clinical studies are huge and uncertain in certain areas, especially we're dealing with pain and such. So there are research investments that are through the FDA, which is good, to understand how to build in vitro in vivo correlations and PBPK models to bridge this bioequivalence gap so that you have more generic products. For example, Dr. Hazem Hassan, who is sitting in the audience, he used to be at the University of Maryland, where he did a study to understand the transdermal absorption and bioavailability using human cadaver skin and to relate it to project in vivo such that you could use in vitro approaches to build or to substitute in vivo studies which are uncertain. It's just that paradigm is uncertain. It's not that you cannot do the study, but you can have false negatives. So this is the type of study, the place where PBPK can be used. And quality management, quality by design, if you have heard, again, you could use this approach to understand which processes in the manufacturing have high impact on the in vivo bioavailability and absorption, and then you can use those approaches to control the quality and release the batches. Special populations. As I said, wherever you cannot access biological fluids, PBPK could be an answer for that. If you can do, for example, we're involved with doing some work in sheep models, because you cannot do this in humans. And then there is a way for you to scale up from animal models to humans using physiologically available data. Pediatrics, this is one single thing I have to say is blown out of proportions by the PBPK community. We do not need PBPK for pediatrics. We should stop doing that. Let me tell you why. I'm already up, I guess, one minute or so. Because pediatrics, you're not going to get a waiver for pediatric PK studies. Let us be very clear about them. So you only need a reasonable estimate of the dose that you want to start for the pediatric study. It's not laser specific. And even if you are off by 10-20%, you're going to learn from that study, and you can adjust the dose for your efficacy and safety trial or for writing labels. So there are some folks who have argued with me saying that the PBPK showed 5% better prediction over allometric scaling. So again, this is not where PBPK is supposed to be used. So I hope I gave you a rational view, or at least it's my biased view of PBPK, where the strengths are and where we should be careful or more prudent in deciding whether that's the tool of choice or not. Thank you very much.